You're at the cutting edge of RNA Therapeutics. I'm your host, John Cook. I'm professor and director of the Center for RNA Therapeutics here at Houston Methodist Hospital. And I'm joined in the studio by my faculty member, Dr. Christopher Brannon.
Thanks, John.
Dr. Brannon, would you like to introduce our speaker?
Sure. Yep. So it's my pleasure today to introduce Dr. John Rossi. He's the Liddell Family Research Endowed Chair, Emeritus Dean at IRO in Manila Graduate School of Biological Science, and Director of Center for RNA Biology and Therapeutics, the Beckman Research Institute at City of Hope. Dr. Rossi has authored over 300 high-impact research publications, pioneering research and therapeutic application of small RNAs, such as ribozymes, siRNAs, saRNAs, microRNAs, aptamers, and more, for HIV and cancer and various other diseases. He developed the first combinatorial RNA-based gene therapy strategy to enter human clinical testing. He's the founding president of the Oligonucleotide Therapeutics Society and a board member of the Society for RNA Therapeutics. He's the co-founder of several biotechnology companies, including MENA Therapeutics, which is developing exciting new small activating RNA therapeutics. And he's the editor-in-chief of Molecular Therapy, Nucleic Acids, and a winner of many awards, most recently the Outstanding Achievement Award from the American Society for Gene and Cell Therapy. So we're really honored to have Dr. Rossi here today, and today he'll be joined by his son, Daniel Rossi, also at the City of Hope, who will be helping present alongside John. And we're joined for discussion by Dr. Martin Requena, who's a postdoctoral fellow in my group, and by Dr. Luis Orlando and Dr. Caterina Castillo, who are postdoctoral fellows with Dan Kiss's group here at Houston Methodist. So welcome, John and Dan, and we'll get started.
John, thanks for joining the show. It's wonderful to have you here with us, and we're looking forward to your talk, but I thought maybe we could just have a little discussion before you embark on your novel inhalable RNA aptamers. John, tell us what got you into the RNA field 40 years ago?
I studied splicing in yeast and bacteria. I studied the RNA processing in bacteria. That wasn't my interest in RNA. Then I went to City of Hope at the AD, interested in ribozymes as therapeutic agents. From there, it's been RNA for me for the last 40 years.
Wonderful. Actually, there was an article in the New York Times today from Tom Cech, as you know, the Nobel laureate, one of your colleagues in the ribozyme arena. And it was actually very interesting to see that in New York Times, an article directed toward the lay public about the promise of RNA therapeutics and how important the field has become for us and the opportunities for new drugs and new therapeutics.
News for the society.
Right, absolutely. Well, maybe we should go ahead and get started with the talk on novel inhalable RNA aptamers targeting SARS-CoV-2, highly relevant and topical. Thanks, John.
Great, thank you. I turn it over to Daniel Rossi now.
All right, great. Thank you so much for the transition. So I'll be walking you through the presentation today. So we'll be presenting on novel inhalable RNA aptamers targeting SARS-CoV-2, and I'll get right into it. So SARS-CoV-2 is able to gain entry to host cells through binding to angiotensin converting enzyme 2, or ACE2, on the cell surface through the spike protein, which covers the surface of its virions, facilitating host entry. Given this, the spike protein provides an attractive therapeutic target for an antiviral agent. Aptamers, like antibodies, are capable of binding to a molecular target with high specificity and affinity, and provide an ideal therapeutic agent in this context thanks to their ease of development and the ability to be manufactured synthetically and at scale. By binding the spike protein with an RNA-based aptamer, the spike protein's interaction with ACE2 can be effectively inhibited, thus preventing viral entry into host cells. As a consequence, Use of a spike protein targeting RNA aptamer as an antiviral or prophylactic can help reduce viral load, mitigate disease progression, and potentially limit transmission of COVID-19. So some quick background on aptamers. To reiterate, aptamers, which are often referred to as chemical antibodies, are short segments of RNA or DNA that can bind to specific targets with high affinity and specificity. Like antibodies, they recognize and bind to their targets through their unique three-dimensional structures. In the context of a therapeutic agent, RNA aptamers are generally more appropriate due to a higher degree of structural complexity and their ability to undergo a wide range of chemical modifications to improve their overall stability and deliverability. One of the most crucial advantages of aptamers come from their ability to generate leads quickly through a process of synthetic evolution. CellX, or systemic evolution of ligands by exponential enrichment, is the process by which we can produce aptamer-based therapeutic leads and consists of four main steps. First, a large, diverse library of random RNA sequences is synthesized, with individual aptamers within libraries being about 80 nucleotides long. This pool is first narrowed down through a process of counter-selection, in which the library is incubated with negative cells that do not express the target protein. Next, positive selection occurs by incubating recovered unbound sequences with positive cells expressing the target protein, after which the target-bound sequences are identified through a nucleic acid barcode-based high-throughput Illumina deep sequencing technique and bioinformatics analysis. This final pool represents therapeutic leads for further testing and refinement. As a technology, aptamers have proven useful both as research tools and as therapeutic agents. To date, the most advanced aptamer-based therapeutics are used in the field of ophthalmology, for which there exist 3 aptamers, including 1 drug approved by the US FDA, Macugen, and 2 in late-stage development. The technology as a whole remains relatively young, and applications across a wide variety of disease indications outside of ophthalmology are being explored in earlier stages of development. So, an important step to note in the development of aptamer therapeutic leads is size truncation. After the successful identification of a high-affinity aptamer to a given target, the next step is truncation. So this involves reducing aptamers to the minimum sequence required to maintain selective binding. And in some cases, this can even increase an aptamer's target affinity. It is thought that the reduction of sequence length may reduce steric hindrance, decrease intermolecular interactions that must be disrupted for target binding, or improve the stability of an aptamer's tertiary structure. Of further significance is manufacturing, with shorter sequences being much simpler to chemically synthesize, with resulting products achieving fewer purification steps and higher overall yields. In the long game of drug development, this results in dramatic cost savings, especially when you move to the GMP scale. So the previous 2 slides provided a generalized overview of the selection and modification of aptamers, and we specifically looked at cell-based CellX, so using an in vitro cell culture. But to begin digging into our generation of aptamers against SARS-CoV-2 spike protein, I also want to quickly touch on protein-based CellX. So the general principle here is the same as that you saw with cell-based CellX. However, in this case, magnetic beads are used for both positive and negative selection. So here, the negative selection is carried out with beads unbound to a target protein, with these bare beads filtering out aptamers in the library with an affinity for the beads themselves, while positive selection occurs on beads bound with the target protein. As with cell-based CellX, this process proceeds through multiple rounds of selection until a highly specific and sensitive set of aptamers is evolved. We utilize this method in order to generate a set of aptamers specific to the COVID spike protein. So these charts represent our final aptamer pools, identified after alignment of the top 100 sequences identified from the initial pool. Frequencies of aptamers are represented after each round of selection, revealing those with particularly high abundance after multiple exposures to COVID spike protein. The one you see called apt1 demonstrated a particular high affinity, and became our candidate for testing against the spike protein of both the original Wuhan variant of SARS-CoV-2 in addition to the more recent Omicron variant. So, as we discussed previously, it's necessary to truncate candidate aptamers to enhance their efficacy, optimize their structure, and increase viability of manufacturing downstream. To this end, we developed a truncated version of APT1, visualized on the left. We compared the binding affinities of this truncated APT1, which we're going to call TAPT1 from here on out, and APT1 to both the spike and Omicron variants of SARS-CoV-2. We conducted binding assays and calculated the KD values, which represent the dissociation constants for each aptamer. The results of our analysis, visualized in the middle of the slide, demonstrated that TAPT1 exhibited a better KD value compared to APT1 for both the spike and Omicron variants, with KD values of 20.73 nanomolars for the original spike and 20.8 for the Omicron spike variant, thus indicating that TAPT1 has a stronger binding affinity to the viral proteins. Furthermore, we aim to confirm the neutralization inhibition by APT1 and TAPT1 against both the spike and Omicron variants of SARS-CoV-2. To assess the neutralization potential, we utilized a cell fusion system provided by Invivagen. Our results demonstrated that both APT1 and TAPT1 effectively inhibited the neutralization of both spike and Omicron variants. The presence of these aptamers hindered the fusion-mediated seep release, indicating their ability to interfere with the viral entry process. So here's a closer look at the neutralization assay I just mentioned that was in the far right figure of the last slide. This assay relies on the transfer of an adapter molecule, MyD88, from a donor cell line to an acceptor cell line expressing an NF-kappa B seep-inducible reporter gene. Cell fusion is then readily accessible in the co-culture supernatant using the seep detection reagent QuantiBlue solution. The aptamer binds with the spike protein and blocks cell fusion, meaning MyD88 doesn't transfer into ACE2 expressed cells and seep doesn't express. Thus, we can easily detect the aptamer's biologic activity in blocking spike protein via reduced seep detection. Having established the preliminary viability of our lead, let's explore the rationale behind using inhaled delivery with aptamers in the context of SARS-CoV-2. The virus spreads easily through respiratory droplets, with a single sneeze being able to produce up to 10,000 droplets, which can contain the virus and potentially infect others. So as we continue to face the challenges posed by COVID, it's important to consider future pandemics as well. The World Health Organization has recently emphasized the need to prepare for the next pandemic involving pan-sarbecovirus, which can spread easily to the airways. To combat the spread of COVID and future sarbecoviruses, inhaled aptamers offer a promising therapeutic approach to target infectious agents within the airways themselves. These aptamers can be designed to target the viral receptors and prevent their interaction with human receptors in the airways. Given the dysregulation of lungs caused by COVID, in addition to the threat of future infectious agents such as sarbecovirus, we wanted to direct our research towards a strategy to treat the airways directly with the SARS-CoV-2 inhibiting agent. As is well established, an overactive pro-inflammatory response might be the underlying cause for the pulmonary complications and subsequent respiratory distress seen in some COVID-19 patients. Notably, following infection with COVID-19, patients exhibited an elevated cell count in their airways, largely due to the increased presence of airway macrophages, T cells, and B cells. Ergo, the initial goal of our research was to provide an agent which could inhibit SARS-CoV-2 in the airways directly, in which we aerosolized SARS-CoV-2 in order to deliver it directly to the lungs in a K18 H-ACE mouse model, which expresses the human ACE2 receptor in mice controlled by the human keratin 18 promoter in the epithelia, including epithelial cells, where SARS-CoV-2 infections typically start in human patients. To this end, we developed an inhalation system to deliver TAPT1 aptamer into mice for the purpose of pre-exposure prophylaxis against COVID-19. This inhalation system effectively covered the mouse nose and efficiently delivered the aptamer with each breath. Our experimental design utilized the system to expose treated mice to a week-long period of prophylactic exposure. with TAPT1 or a control aptamer delivered intranasally at days -6, -3, and 0 at 20 nanomoles. Following this treatment window, mice were challenged with SARS-CoV-2 infections, inoculated intranasally on days 1 and 5. So here you can see the change in mouse weight over time, highlighting a reduced weight loss in the TAPT1 group relative to the control group, which provided preliminary evidence of a potential therapeutic effect of TAPT1 in murine COVID exposure using this model. Delving into our key data, we detected SARS-CoV-2 in lung, heart, spleen, kidney, brain, and the pancreas. While a slight reduction was observed in lung tissue, contrary to our initial expectations, and more substantial reductions were observed in the heart, spleen, and kidneys, a remarkable reduction was observed specifically in brain and pancreatic tissue in treated mice. The absence of live virus in the brain and the pancreas really underscored the potential of inhaled TAPT1 in thwarting SARS-CoV-2 in these tissues. So collectively, these results illuminate the potential therapeutic role of this aptamer against SARS-CoV-2 infection, especially when it comes to those two organ systems. I think most intriguing and unexpected of these results were the results that we saw in brain tissues. So as you can visualize on the left, the viral load in the brain was dramatically reduced between the treated and control groups. Immunohistochemistry staining highlights this protective effect even more dramatically, with clear evidence of infection in slides taken from mice receiving the control aptamer relative to healthy tissue in the treated animals. While this result is surprising in light of our initial goal to produce an inhibitory agent which provides a protective effect in lung tissue, it also provides striking new possibilities in the treatment of COVID-19. So from the first weeks of the COVID-19 pandemic, it was clear that many patients suffered from neurological symptoms, ranging from headaches and brain fog to strokes and paralysis in more dramatic cases. Now, while it's generally thought that these symptoms are caused indirectly by inflammatory effects triggered by the immune response to SARS-CoV-2 infection, the possibility of direct infection, as observed in this model system, has not been entirely ruled out in humans. Given this, a prophylactic agent which could inhibit COVID in the brain or surrounding tissues may hold therapeutic value in treating cases of COVID-19 with neurologic symptoms. Furthermore, these data suggest that nasally administered aptamer viral inhibitors may hold potential to treat other viral infections of brain tissue. So, to draw some conclusions, we have found that aptamer-based inhibition of SARS-CoV-2 spike protein is an effective method to inhibit COVID infections in the brain and pancreas. This inhibition was achieved through the prophylactic administration of a spike protein targeting aptamer in the week leading up to viral challenge. Further investigation is required to elucidate the activity of this agent in other tissues, such as the liver and heart. Contrary to initial expectations, the nasally inhaled aptamer did not provide a significant benefit to the infection of lung tissues. And further investigation is required to determine why inhibition was more significant in other tissues relative to the lungs. The precise mechanism of action of the anti-spike protein aptamer is not fully understood at this point, especially in regard to its protective effect in brain tissue. Despite this, these data offer exciting new research possibilities in other infectious diseases of the brain, suggesting the use of nasally inhaled aptamers for this and other purposes should be explored more generally. So, future directions include whole organism biodistribution assays of TAPT1 to better understand how the agent distributes through the body when administered nasally, hopefully elucidating its improved biologic activity in some tissues relative to others. It is also worth exploring additional delivery mechanisms, such as oral inhalation, to see if it's possible to establish a protective effect in lung tissue by other methods, as was originally intended in this study. The mystery as to TAPT1's efficacy in brain tissue is particularly intriguing and requires much closer examination to determine if aptamers might represent a more generalized strategy to treat viral brain infections. Further studies are required to determine the durability of the prophylactic effect TAP1 is demonstrated in brain and pancreatic tissues to determine exactly how long after treatment they can resist viral challenge. Furthermore, the specific mechanisms by which this effect was observed in brain and pancreas, but not other tissues, remains to be elucidated. So moving into acknowledgements, these include Min Sun, who is a staff scientist in the Rossi lab, as well as former lab members Jiwa Zhou and Alberto Herrera. Funds for this project were provided by an NIH R21 to the Rossi lab. Additionally, our CRO partner BioQual, located in Rockville, Maryland, performed all SARS-CoV-2 inhalation challenge assays for this study. Thank you for your time today, and I'm now going to pass things back to Dr. Rossi so he can field any questions you might have.
And Dan, that was fascinating, really fascinating. One of the questions that I had was regarding the your interesting effects on brain tissue. You showed some immunohistochemistry that suggested a reduction in SARS-CoV-2 in the brain of those animals that were treated with the nasal aptamer. Let me ask you a question about that. Where was SARS-CoV-2 in the brain? Was it in the brain parenchyma, or was it in the endothelial cells? Where was it ending up?
We just started to do some pathology. Looks like endothelial cells, blood vessels in the brain.
Yeah, that makes sense to me, that it would be the endothelial cells it was ending up in. And that certainly could cause activation of the endothelial cells and an increase in permeability, which some have blamed for the symptoms of long COVID, the COVID endotheliopathy. Fascinating, really fascinating. When you embarked on this study, did you, I'm sure you went through the calculations of how much aptamer you'd have to deliver to prevent an active infection. I guess my question is, how many multiplicities of infection do you need? In other words, how many active variants do you need to induce an infection? I guess then you can backtrack from that number to figure out how much aptamer you'd have to deliver.
Actually, that day was done by BioQual. They had the estimates for the infected numbers.
Yeah.
I don't recall the actual number though.
Yeah.
But we used 10 nanomolar for our aptamer. That was the amount given each mouse. I should say there were 26 mice in this study, 13 females, 13 males, equally divided between control and functional aptamer in each case.
Great, thanks. The aptamer must get absorbed in the nasal mucosa into the general circulation. Do you have any thoughts about the biodistribution? Does it apparently it gets to the brain quite well, at least the brain endothelium, the aptamer?
Actually, we have this under investigation now. We don't really know exactly how the aptamer protects in the brain.
Yeah, great. Questions
I think it
from
might be through the ophthalmic nerve.
Let me turn to our students for a moment and see if they have any questions.
All right. Hi, Dr. Rossi. Can you hear me? This is Katharina.
I do hear.
Yes. So I'm curious about the CellX assay, the cell-based CellX assay. So what specific types of cells did you use for that? So one can imagine that different types of cells can lead to the modifications of the target protein, and that could also lead to the non-specific binding of your potential target aptamers.
Yes, good question. Actually, we use purified protein for the original CellX. Then the cell lines that we use express the protein. They were HeLa cells, I think.
Other questions from our trainees?
Yeah, Luisa, I think you're muted.
Louisa, you might be muted. Yeah, it appears that you're muted.
Yeah.
She can't unmute.
No,
Oh.
no, because yours is not unmuted otherwise.
Hi, great talk. I was wondering about the original Celex where you used the protein to identify the aptamer. The schematic had just empty magnetic beads as your negative control. Did you happen to use any other proteins that you didn't want it to target in that negative selection?
Actually, that's a good question too. They actually used COVID-1 spike protein as a negative to control. Just to get some that just binds the proteins in general.
Other questions, comments?
Martin.
Go ahead, Martin. You're muted.
Martin, you're muted.
You're still muted, Martin. While we're working on that, just it's very curious that there's a specificity for the pancreas too. So pancreas and brain, is it because there was some affinity of the aptamer for the pancreatic endothelial cells and the brain endothelial cells? Are there some surface markers being expressed there that might account for that selectivity?
That's a very good question. I wonder if it has to do with the H2 receptor density on pancreas.
So there could be.
Actually, I'm not really sure why the pancreas and the brain were the two protected targets.
Right. So it could be that there was greater preference for the pancreatic ACE2 receptor, brain
Yes.
ACE2 receptor. It'd be interesting to understand what those differences are between the different endothelial different circulations. Martin, are
Exactly.
you ready to talk to us?
Martin's device might be disabled.
He's still having some problems with his.
I guess one question I had is you did your CLX experiment against the RBD protein itself, but I was curious if you considered maybe doing a CLX experiment against the ACE2 receptor itself. I think some folks have used ACE2 blockers or ACE2 inhibitors as an alternative therapeutic for SARS-CoV-2.
Yes, we thought about that. We're concerned about the role of that receptor in normal human function.
Yeah.
So that's why we chose the virus instead.
Correct. Yeah. So it's better to target the virus in this case because the ACE2 receptor has an important function for endothelial vasodilation, I think.
And I guess how do you imagine this in practice? This would work as a prophylaxis. Folks would sort of carry a nebulizer around, and if there was some sort of a situation where they feared infection, they would use it as a prophylaxis, or how do you imagine this translating?
That would be the best way to do it.
Okay.
We have not really considered that commercial use yet.
Yeah.
Just look at the basic science of this whole thing right now.
How long is the on rate? How long is it going to be effective once it's sprayed into the nasal mucosa? I guess that would be another question. How often would you have to use it?
Right.
That's a good question. That's something we have to study still. The duration of effectiveness, we don't know right now.
Yeah.
All we know is that 5 days after we give the aptamer, we're doing the detection, it still protects.
Oh, that's interesting. So 5 days after administration, you still got some protective effect. I mean,
Yes.
You could see it being very useful if you had to, take a plane flight, for example, or you had to be at a big conference like I was last week, and now I've got this upper respiratory tract infection. I could have used your aptamer Do you think there'll be something a little bit more broad-based, John, that we could use that would prevent a number of viral infections?
That's the hope, that to find the most effective approach to afterbirth target gene.
Yeah, interesting.
Yeah, and can you hear?
Yeah, we can hear you.
Yes. So yeah, just curious also about so these are aerosolized aptamers, correct? RNA aptamers. Are they combined with anything else? And I'm just curious about stability or long-term storage.
Saline solution only.
Okay.
So they're two-part floor backbone, so much stable against nucleases.
Oh.
Great. Okay.
What's the best strategy for assessing biodistribution of an administered aptamer like this? What are you guys planning to do for that?
My idea is to do PET imaging. They see where they get the most definitive information. We're working on that right now. Just got a student working on it.
So you have for PET. I guess that would be a radio-labeled agent, or it would be.
Yes. Yeah. Right. Exactly.
Yeah. Okay. I think Martin is. Have you corrected your mute, Martin? Can we hear you? I guess not. Sorry about that.
See you, Martin.
If you put your question in the chat, maybe we could ask the question for you.
Are there any questions in the chat from the audience?
I don't see any yet.
Okay.
Well, while we're waiting for Martin, I want to thank you, John Rossi, for joining our Society for RNA Therapeutics. You're on the board of the Society for RNA Therapeutics, and we have some great people like yourself, other great scientists on the board. Bruce Sullinger from Duke is another one of your colleagues who does a lot of aptamer work. We have Leo.
He's actually a good friend of mine.
Yeah, yeah.
How do you explain the SARS-CoV-2 in the brain? Is it the aptamer itself gets? What we believe is happening after it blocks the virus and infecting in the endothelial cells in the brain? That's how we explain it. That's the only explanation I have right now.
Yeah. No, it does make sense, too. If you can block the endothelial activation and the increased permeability that results therefrom, that could have a beneficial effect on long COVID and the encephalopathy that occurs.
I think Louisa has another question.
Louisa?
Hi, yes. I was wondering about the truncation. As you can imagine, cutting off too much might prevent binding.
Exactly.
Is there a way that you specifically decided on what to truncate and what to leave so that you have the smallest possible?
First, structural analysis, trying to look at what might be hanging off from secondary structure. It's not science yet, it's just kind of a guessing game.
Yeah, along that line too. So I assume for the Celix, it's more on sequence dependence, right? So how did you get away with the structure, possible structure effects? Because once they form structures, they can also bind, but non-specifically. So how did you get away with that?
Actually, we believe it's like a tertiary interaction of the aptamer with secondary structure, with tertiary structure with a target, like an antibody binds to a protein, same principles. So this time it's just nucleic acids. So it's kind of something that's hard to model. Nobody's really modeled aptamer binding yet. Something I would like to do in the future, do some crystallography, see what's actually going on.
I guess sometimes you just have to do the experiment, right? I mean, there were so many unknowns, uncertainties when you embarked upon this with the aptamer, and you have to do the experiment. Is it how many multiplicities of infection can create a problem for the patient? How many aptamers do you need? How much do you need? The molarity of it to reduce the infection. Distribution
Yes,
of the aptamer. All of these things were kind of unknowns.
Right. It was kind of a dive into the dark waters of biology.
Into the dark waters of biology. That's absolutely right. Well, we really enjoyed the chance to talk to you, Dr. Rossi. The work looks very promising and very interesting, intriguing, the effect that you saw on the brain endothelium, preventing the endotheliopathy that can occur with COVID infection. Thank you for that, and keep up the great work.
Thank you so much for inviting me. We have a publication on my archives. If you want to just email me.
Wonderful, wonderful. I look forward to that. Well, thank you, John, for joining us, and your son for joining us on the cutting edge of RNA therapeutics. And thank you all to attended today. I'm your host, John Cook, and Christopher Brannon, Dr. Brannon, and I enjoyed being on the show with you today, Dr. Rossi. Bye.
Thank you very much. Bye.
Yeah. Thanks, Dr. Rossi. Bye.